Assalamualaikum and good afternoon. My name is Nur Ajana Faisha binti Saira and my metric number is 102805. I'm from a plant science student from Faculty of Resource Science and Technology. Today I am honored to present my final year project proposal entitled Isolation and Morphology Identification of fungi associated from Oryza sativa leaf disease. My supervisor for this project is Dr. Margaret binti Sidi. So, let's move to the next. To begin, let us look at the significance of our target crop. Rice, significantly known as Oryza sativa L, belongs to the family Poaceae. It stands as the second largest cereal crop produced globally, acting as a primary staple food for more than half of the world's population. Because it thrives exceptionally well in tropical and subtropical climates, it is central to Malaysia's agriculture sector. Beyond being an essential source of carbohydrate and daily energy, rice plays a foundational role in maintaining national food security and driving our agrarian economy. Despite its critical importance, rice production faces several biological threats. Rice plants are highly susceptible to several devastating fungal pathogens. Among the most common damaging leaf diseases are rice blast, brown spot, leaf blight, and narrow brown spot. These fungal pathogens aggressively attack vital parts of the plant, including the leaves, stems, nodes, and seeds. When severe infections take root, they drastically reduce photosynthetic capability, leading to significantly lowered grain yield and compromised crop quality, which directly impact farmers' livelihoods. So, this brings us to the core issue. Rice disease remains an ongoing major agriculture hazard. Historical data spanning from 2009 to 2021 indicates that more than half of the rice fields across Sarawak exhibited moderate to severe disease intensity. Yet, despite this widespread severity, very few studies have specifically focused on comprehensive fungal identification within the local ecosystem of Sarawak. This has left us with a critical gap, a lack of detailed fungal identification and morphology categorization data. Without this baseline knowledge, farmers and scientists cannot achieve accurate diagnosis, which is absolutely vital for design targeted, proper disease management strategies. To address this gap, my research is guided by three main objectives. First, to determine the frequency of fungal infection linked directly to rice leaf disease. And for the second one is to evaluate the severity and distinct symptoms of this rice leaf disease caused by fungal pathogen. And the last one is to assess the potential variety and diversity of the fungal population interacting with contaminated rice leaves. So now, according to literature, the Poaceae family is incredibly diverse, encompassing over 10,000 distinct species, making it the most commercially significant plant family for global food security. This family includes the world's primary staple crops, which are rice, maize, and wheat. Furthermore, prominent studies by New and Gonzalez confirm that complex fungal complexes drive these leaf lesions. In control, pathogenicity experiments such as those conducted by Iman and colleagues in 2022 Inoculated plants consistently display dark or brown patches, while control plants remain completely symptom-free, proving a direct causal relationship between the targeted fungal pathogens and the visible disease symptom. So, uh, for the methodology that I will use to achieve this objective is this is the workflow is divided into systematic laboratory stage. For the first one is in sample collection. I will gather infected rice leaves showing visible symptoms from selected fields, label them precisely by that allocation, and transport them safely to the UNIMAS laboratory. Next, I will perform the spore drop and isolation technique. The infected leaves will be cut into small segments, surface sterilized using 1% Clorox for exactly 1 minute. This is to eliminate several phytic contaminants, and then rinse 3 times with distilled water. This tissue segment will be placed onto agar media and incubated to allow fungal growth, allowing us to eventually isolate single spores under the microscope for peer culture propagation. Next, once peer culture are established, morphological characterization will be carried out using two probing approach, which are macroscopic and microscopic. For the macroscopic, it will observe and assess visible colony traits, including colony color, texture, shape, elevation, and overall growth rate on the petri dish. For the microscope, it will observe and will develop into circular structure. Under the microscope, I will closely analyze hyphae structure, margin appearance, spore morphology, conidiophore structure, and distinct sporulation patterns to accurately classify the fungi. Throughout this research, we confidently expect the successful isolation of fungal pathogen from infected leaves, resulting in clearly differentiated fungal species with distinct colony morphologies. Ultimately, these fungi will be tentatively identified to their genus or species levels with clear microscopic structure documented. As shown in my Gantt chart, the timeline spans from proposal preparation in late 2026, progressing throughout active laboratory isolation, inoculation, and profiling across early 2025, and after that, culminating in final report writing and submission by June 2027. So I think this is all from me. Thank you for your cooperation.